Welcome to Neuromatch Academy. We have prepared for you a very exciting program for the next 3 weeks, during which you will learn about state-of-the-art approaches to modeling and model-based data analyses. My name is Gunnar. I'm a co-founder and co-organizer of Neuromatch Academy and a founder of Cosmo. I'm a professor at Queen's University in Canada and I study sensory motor control in my lab. And in my spare time, I'm also big time into brewing beer. Scientific progress is made through an interplay of experiments, data analysis, and modeling. Neuromatch Academy is all about data analysis and models. And before we can fully dive into what specific models can do for us, we first need to understand a little bit more what models are and how we go about modeling. These will be the topics of the first 2 days, during which we will slowly introduce you to models. That will be followed by model fitting on day 3, how machine learning can help us on day 4, and how you can do all this when you have high-dimensional data on day 5. Then in the second week, we will dive much more into crucial models and approaches that span much of systems neuroscience. And in the third week, we will cover biological and artificial neural networks, as well as how we can get causal insight. So today, we'll talk about what models are, different types of models, and what they can do for us. So why are models so helpful for scientific progress? Well, they can do many different things for us. For example, they can synthesize knowledge. In other words, they can summarize a large body of findings into a rather small or compact description, which is the model. They allow us to identify hidden assumptions, hypotheses, unknowns. In fact, once we have to start modeling, once we think about quantifying our models and writing down the mathematics, very often we come across findings or facts that we need that are not available or that we haven't thought of. Models can provide mechanistic insights. That is one of the main reasons why we might want to have models, is that we cannot measure certain things, but we can infer them if we can capture a mechanism that the data underlies, for example. We can retrieve latent information. So latent information is information that is not directly present in the data. But with a model, we could potentially extract such latent information. And I will show you an example in a bit about this. Models can be a test bed for medical interventions. If you have a working model of the brain and you can somehow manipulate that in a way that is interesting for medicine, then you can ask essentially what this manipulation does to the brain. Models allow us to design useful experiments, for example, by making quantitative predictions. And this can be particularly important if these experiments involve animals that sacrifice their lives for us. So instead of doing arbitrary experiments that follow simply our inspiration or mood of the day, we can focus on only conducting the important experiments that make meaningful predictions for our models, or experiments that allow us to test between different hypotheses or between different proposed models. Finally, models can inspire new technologies or applications. And again, I will show you an example about that, but there is the whole field of neural networks, for example, as the name already indicates, that has been inspired by, at least roughly inspired by how the brain works. Okay, so specifically, let's look at 3 different models. So first, let's look at the popular cosine tuning model of neuronal activity, the famous Hodgkin-Huxley model of single neuron spike generation, for which they got the Nobel Prize, and the very fashionable reinforcement learning model. And that should give you a bit of an idea. At the end, we'll come back to the list of advantages and for different models. So that should give you a bit of an idea what different models can potentially do for us and why they're useful. So here we go. Cosine tuning refers to an often observed spatial response profile of neurons with respect to a stimulus or a movement that can be well approximated by a cosine function. Here, for example, we have 5 repeated movements of a monkey. She moves in each of the 8 movement directions from a central starting point. The neural activity recorded from a primary motor cortex pyramidal cell is shown as a raster plot. Each line corresponds to one movement instance, and each tick represents one action potential. Around the movement onset time, denoted by the vertical bar, this neuron's activity is modulated from either highly active to silent, depending on the movement direction. So you can plot the average rate of spiking as a function of movement direction, and this is well described by a simple model containing only one equation. And this is the equation. The equation takes the firing rate f as a function of the movement direction s, subtracts the baseline firing rate f0, and divides that difference by the maximum firing rate of that particular neuron. So now you have a normalized firing rate, and that across the different stimulus, the different movement direction is well approximated by a cosine function that peaks at this neuron's preferred movement direction, SP. So why is this cosine tuning a helpful model? Well, it provides a really nice compact summary, a description of the data. It generalizes across movements, so you can essentially well predict what would happen if you recorded in the same cell for movements that were not part of the 8 different movements that we have currently seen, but somewhere in between, let's say. The cosine tuning model also applies to sensory stimuli, and we can use this for applications. For example, to build a brain-machine interface for prosthetics, right? So if we were to be able to quantify each neuron's cosine tuning function parameters, and then use that to decode the movement intention of the brain, that could potentially work fairly well. However, this is a purely descriptive model. There's no consideration about how or why cosine tuning arises in the brain, and therefore it provides scientifically limited insight. Okay, now let's look at a very different model. In 1939, Alan Hodgkin and Andrew Huxley recorded the intracellular potential of the giant axons of the squid. This resulted in a first recording of an action potential. Unfortunately, then the Second World War happened and both were busy with military duties. But after the end of the war, they got back together to understand what mechanisms led to the depolarization at the beginning and the hyperpolarization at the end of the action potential. Today we know it's due to the opening and closing of specific ion channels. When a threshold membrane potential is reached, first sodium channels open, raising the potential. That's the panel number 2. That leads to potassium channels to open, so the rising of the potential leads the potassium channels to open, and the sodium channels to close. That brings the potential back down. So that's panel 3. And there's an overshoot in the potential because potassium channels close slowly, resulting in a refractory period where spiking is suppressed. That's panel 4. So Hodgkin and Huxley knew that there was electrical currents involved, because that's essentially what they measured with their electrodes. And thus they used basic electricity theory to formulate their model. So the model was has the form of a relatively simple first-order differential equation that abstracts the axon or neuron by a capacitor and describes the change of electrical potential between intra and extracellular media as an equilibrium equation involving sodium ions, potassium ions, permeability of the membrane through leakiness, and external currents. So the individual ions and leakage channel reversal potentials are represented as batteries and the corresponding channels as resistors. And then they introduced ion channel opening and closing variables N, M, and H as first-order differential equations and found that they had to take some power of those variables to accurately describe channel opening and closing kinetics. Now, why is the Hodgkin-Huxley model helpful? Well, Hodgkin and Huxley provide a mechanism for the generation of action potential, right? It's the opening and closing sequences of the different ion channels. The model synthesizes large amounts of neural data and describes variables that are not easily measurable. So these latent variables, right? So for example, the probability of channel opening and closing, or the currents going through each of those channels. And Hodgkin and Huxley also, or their model also allows for studying effects of interventions. So for example, we could study how a potassium channel blocker would affect spiking behavior of their neurons. And you can make real predictions. So for example, you can look at the conditions that control the timing of the action potential onsets, so the threshold or refractory period. But we really have no idea why ion channels open and close, what the molecular mechanism is that makes them open and close depending on the difference in voltage between the intracellular and extracellular medium. All right, so here's yet another very different model that's called reinforcement learning. So while the previous two models tried to capture spike statistics and spike generation mechanisms respectively, reinforcement learning asks how agents or people or animals should be acting to maximize overall reward. So imagine an agent trying to get out of an ordered labyrinth environment. The agent will take an action, so a movement, right? And then observe the result. That is the new position that the agent is now in after the movement is completed, right? So the new position that the agent is in the environment, and a reward if it got out of it. So if the movement or the action was positive, it will receive a reward. So if the agent receives a reward, then that means that this movement was a good choice. And the agent will thus update its policy, meaning it will update the rule. So the policy is the rule by which it chooses movements when encountering the same situation or the state. So in that way, the agent will reinforce actions with rewarding outcomes and thus learn how to move optimally through the environment. So why is reinforcement learning such a nice model? Well, it provides a normative benchmark for what is best in theory. So normative essentially means exactly what is best in theory given certain assumptions. And that includes predictions for optimal behavior. So outside of what has been tested experimentally, for example. Now, the reinforcement learning model also allows us to synthesize large amounts of behavioral and also some neural data. It describes variables that are not easily measurable, so these latent variables that are hidden, that we cannot directly measure in experiments. For example, things like the reward prediction error. And reinforcement learning has inspired new technology. In fact, reinforcement learning is central to modern artificial intelligence. But we have no idea how the brain achieves it. How is reinforcement learning implemented in the neural circuits of the brain? So let us summarize again why we thought these three models were great. And we'll go back to our list from before why we think models are great. So we talked about knowledge synthesis. And in fact, all three models, cosine tuning, Hodgkin and Huxley, and reinforcement learning, are able to synthesize knowledge by capturing large amounts of data. Hodgkin and Huxley also allowed us to identify hidden assumptions, hypotheses, and unknowns, things like discovering ion channels, for example, and provided mechanistic insight. So why does the action potential actually occur, right? Well, it's due to the opening and closing sequence of the ion channels. And then Hodgkin, Huxley, and reinforcement learning models allowed us to retrieve latent information, things that we cannot directly or that are difficult to directly measure. Like in Hodgkin, Huxley, that was, for example, the opening or closing of the channels. And in reinforcement learning, that was the reward prediction error. Hodgkin and Huxley could also be used as a test bench for medical interventions. Remember, we could be applying a potassium channel blocker and see how that affects spiking. And the Hodgkin, Huxley, and reinforcement learning models could allow us to design new useful experiments through quantitative predictions they're making. And then finally, we said that another benefit of models could be that they can inspire new technologies or applications. And we said that cosine tuning can do that through, for example, helping us designing brain-machine interfaces, and also the reinforcement learning model. Remember, AI. Okay, so now while we have now seen some models and gotten a bit of an appreciation what different kinds of models can do for us, there's one important thing that I have not told you yet. And that is, what are these things that we call models? And the answer is actually quite simple. Models are an abstraction of reality. As such, models are partial, imperfect descriptions of the universe that are developed by science for our understanding of the universe that is otherwise too complex to grasp by the limits of the human mind. And that, of course, immediately raises the question, how do I know what the right level of abstraction is? The simple answer is, keep it as simple as possible, but as detailed as needed. This is often also known as Occam's razor. In fact, William Occam famously said, entities should not be multiplied without necessity. In other words, shave away unnecessary elements. Now, practically, the right level of abstraction is largely determined by our exact question, the hypotheses we would like to test or implement with the model, and our model goals. And that is what tomorrow's day will be devoted to, and you will learn all about how this works. But ultimately, we want models to empower us scientifically, right? To give us something that we cannot directly get from the data. So there's two big things that we cannot directly get from the data. One is understanding. It's some insight that is not directly accessible by the experiments or the data. And control in the form of interventions, either experimental or clinical interventions, for example. And of course, each model requires to be validated, right? And model validation is essentially done through experiments. In other words, a model is a hypothesis. A model is a precise mathematical instantiation, a mathematical description of a hypothesis. And as such, experiments perform hypothesis testing. And if you want to compare between multiple models or multiple hypotheses, that is called model comparison. Okay, and that brings us back to experiments and data and how they both mutually inform models. And that is pretty much the cycle of discovery, right? You start off with a phenomenon or question, something that's unknown. You think about it, you have an idea, a hypothesis, and then ideally you want to build a model to quantify, to instantiate this idea, this hypothesis that you have in your head. This model can then make predictions, so you get essentially a model outcome that you can compare to your original phenomenon, but often you also get predictions for which you do not currently have data, and so you want to run new experiments to collect that data and test those predictions, right? So therefore, models really inspire experiments, but vice versa, of course, it's the data that inspires the models. And the data, most importantly, constrain the models and provide new questions we would like to understand or be able to answer. Okay, so I showed you 3 specific examples of models, but there's a whole universe of models out there. So one way to classify models would be to just look at the scale of the system they're describing. So here is essentially a hierarchy of levels of model organization that goes all the way from the multiple people interactions to the single brain, to the individual systems, so multiple brain areas working together, to individual brain areas or maps, to networks of neurons, to single neurons, to single synapses, to the molecules in a single synapse. And some models bridge multiple levels of organization. So for example, you can have models that are in the form of neuronal maps that describe behavior very well. Or you can have a model that bridges a network to the system. Such models could be very useful, for example, in understanding, let's say, brain imaging signals. Now, how do the models do that? Well, they do that, for example, by providing relationships between different brain signals from different levels of abstraction, or how such brain signals relate to sensation and behavior. And in fact, much of what we will be talking about during Neuromatch Academy is exactly this. What are the modeling and analysis techniques that can give us insight into how the brain reacts to its environment and how it decides on and generates behavior? Now, it turns out that this universe of models can be summarized in a hierarchy of 3 different levels of generality of models. So at the lowest level, we have the descriptive or what model. They provide a compact summary of large amounts of data and essentially ask the question, what is it that we want to describe? And you will see such a model in tutorial 1. The next level, that is a little bit more general, is the mechanistic or how level. And it shows how neural circuits perform complex functions. And we'll see one of those in tutorial 2. And the highest level, or the highest level of generality is the interpretive or explanatory level, the why level. And the why level is concerned about explaining why the brain does something, for example, because it's optimal. And you will see such an example in tutorial 3. So in order for you to appreciate the differences between such models, you will be exploring them in the tutorials, as I said. We will see how different questions will lead to very different models. So as you all know, neurons communicate by sending action potentials to other neurons. And one striking feature of those is that the time interval between 2 action potentials emitted by a neuron follows a very specific distribution. So as scientists, we're detectives, right? And we can ask different questions about that. We can ask, what is a good description of that distribution? Or we can ask, how is this distribution generated in neurons? Or we can ask, why is this the best thing for a neuron to do? So one thing we can ask is, what distribution best describes the interspike interval? And that's an interesting question because it could tell us something about the brain's physiology. For example, For most parts of the nervous system, you can capture this distribution by a Poisson process. A Poisson process is what generates a Poisson distribution, which is part of the exponential functions family. If you include a Gaussian refractory period to capture the fact that once spiked, neurons cannot easily spike again right away due to being hyperpolarized, then you can fit interspike intervals well. It turns out that a Poisson process is independent of history. This tells us, as scientific detectives, that we don't have recurrency, meaning neurons do not project back to themselves, which would produce a history effect. So in tutorial 1, you will develop a simple descriptive model of the interspike interval and learn what kind of scientific questions this model can answer. Then in tutorial 2, we will impersonate a very different scientific detective and ask what mechanism gives rise to the interspike interval distribution. How does it happen? Remember from the Hodgkin-Huxley model that neurons have a capacitance? Well, that means input currents will accumulate charges on this capacitor, which is akin to integration. But is that integration enough to generate the interspike interval distribution? Well, you will see that excitation-inhibition balance is key, and that such a model can answer potentially very different questions than the one from tutorial 1. In the third tutorial, we will put on yet another scientific detective hat and ask why this particular distribution of the interspike interval is the best thing a neuron can do. In fact, you will see why this particular spike distribution is the optimal thing for a neuron to do if it wants to maximize information transmission with a given or constant number of spikes, let's say in a given time window. And finally, we will have a group discussion to ask what you think the best model is and why you think so. And again, you will learn details about these models later during Neuromatch Academy. Here, it's important to appreciate how diverse models can answer different questions and how different questions lead to different models. And with that, I hope you will enjoy discovering for yourself how different models that describe action potentials can answer different, meaningful scientific questions. Enjoy.